Good morning everyone, I am Leslie Lorenzo and I will be discussing the assigned mechanical parts of the microscope. First, what is mechanical parts of the microscope? Mechanical parts is structures that supports and operates the whole microscope. So now let's move on to the mechanical parts, which we have the base. Y or U shape stands that support the microscope. Next, we have the pillar. Metal attached to one end of the base also support the said microscope. Next, we have the handle or the arm. Third portion arise from the pillar used in holding the microscope. And we have the inclination screw or what we call pivot at the junction of the pillar and the handle used to tilt the microscope. And lastly, we have the body tube, cylindrical structure arising from the handle, holds the desk shield and nose piece with objective at the end.
Good morning guys, I'm Jordan Ramon and my report for today is all about mechanical parts of microscope. Draw tube, upper and smaller end of the body tube, bearing the eyepiece or ocular. Function. Carry the ocular, it can be adjusted to control tube length and so effect correction for the objective lens. Revolving nose piece, circular structure at the lower end of the body tube to the objective are attached. Function, holds two or more objective lenses and can be rotated to easily change power. Adjustment screw, of wheel attached on either side of the body tube. Dust shield, thin circular structure of the lower end of the body tube protects the objective and specimen from dust. Function, binds fine particular matter by electrically charging the particles. Coarse adjustment, bigger wheel to adjust the LPO in focusing. Function, allow for quick focusing by moving the objective lens or stage up and down.
Princess Diane Pilien, our report for today is all about the parts of the microscope. First, fine adjustment knob, smaller wheels for delicate focusing for high-power objectives and oil immersion objectives, making specimen more clear and vivid. Stage, square platform with an opening at the center where slide is placed during focusing. Stage clip, paired structures on either side of the stage to hold the slide in place. Mirror rack, below the stage and attached to the pillar, holds the mirror. Condenser, lens found immediately beneath the hole on the stage, used to concentrate light rays in the specimen.
Good day to everyone. I'm Catherine Omabini. Today, my topic is to define illuminating and magnifying parts of microscope, mainly lenses and glass parts. So, the one is ocular or eyepiece, the nearest lens to the viewer's eyes to peep during actual focusing. Next is mirror. Below the stage, near the base with concave or plane surfaces, used to collect and direct light. Next is iris diaphragm. Below the stage, with several arranged circular blades, regulates a central opening to decrease or increase the reflected light on the object. And last is objective. Tube-like structures attached to the nose piece. In the objective, we have a low power objective, high power objective, oil immersion objective. Low power objective, marked as 10 times or 6 times shorter and forms the general outline or wider portion of the object. The color of this objective is color red. And then high power objective, marked as 45 times or 43 times. In the laboratory is 40 times. Longer form bigger images. Used to examine living microorganisms in drops of water. The color of objective is color blue. Oil immersion objective, marked as 100 times or 90 times. Highest degree of magnification and used to examine stained smears of microorganisms. The color of objective are color yellow.
Compound microscope. It contains more than one magnifying lens and primary tool of microbiologists used to examine and study microorganisms. There are different types of microscope, and one of them is bright field microscope. The object magnified appears dark against bright background. Another type of microscope is dark field microscope. Because there is no direct background light, the specimen appears light against a black background. This is ideal for studying unstained or transparent specimen that absorbs little or no light, and useful for examining external details like outlines or surfaces, and used to view spirochetes. Another type of microscope is phase contrast microscope. Frits Zernike is a Dutch physicist in 1934. Phase contrast microscopy is based on principal differences in refractive indices and light waves passing through transparent objects. Phase contrast microscope has a contrast-enhancing optical technique to produce high-contrast image of transparent, which includes thin tissue slices, living cell in culture, subcellular particles like nuclei and organelles. Diffraction is the scattering of light rays as they touch a specimen's edge. When the two sets of light rays are brought together, they form an image of the specimen on the ocular lens, relatively light or in phase, through shades of gray to black or out of phase. Phase contrast microscope and its parts: transmitted light biological microscope, digital camera system, observation, phase plate, condenser annulus, condenser, specimen, objective, direct or surround light, image plane, and diffracted light. Types of microscope and their visualization. First is bright field microscope. Second is dark field microscope. And third is phase contrast microscope.
The differential interference contrast microscopy. This is similar to phase contrast. It uses differences in reflective indexes. It also uses two beams of light instead of one. Prisms split each light beam, adding contrasting colors to the specimen. The resolution is higher than phase contrast, and the image is brightly colored and appears nearly 3D. The fluorescence microscope. The fluorescence microscope make use of ultraviolet light and fluorescent dye called fluorochromes. The specimen appears to shine against a dark background. It uses a higher intensity of light source, and this in turn excites a fluorescent species. The fluorescent species emits a lower energy light of a longer wavelength, which produces the magnified image instead of the original light source. Fluorescence microscopy can be used to visualize structural components of small specimens such as cells and to detect the viability of cell populations. It may also be used to visualize the genetic material of the cell, DNA and RNA. Mycobacterium tuberculosis can detect by the appearance of bright yellow organism against a dark background. And the Bacillus anthracis appears apple green when stained with fluorescence isothiocyanate. The next is confocal microscopy. Most confocal microscopes are used in conjunction with computers to contrast three-dimensional images. This is used to evaluate cellular physiology by monitoring the distributions and concentrations of ATP and calcium ions. And the last is the two-photon microscopy. It is a fluorescence imaging technique that allows imaging of living tissue up to 1 millimeter and 1000 micrometer deep. The two-photon microscopy can track the activity of cells in real time.
Electron microscope. Electron microscope utilizes a beam of electrons to create an image of the specimen. The electron beam serves as the source of illumination, and magnets are used to focus the beam. The first prototype of this microscope was built by the German engineer Ernst Raska in 1933, which had a resolution power up to 15 nanometer. Modern electron microscopes are capable of magnifying objects up to 2 million times. It also used to visualize viruses and subcellular structure of the cell. There are types of electron microscope. First, the transmission electron microscope or TEM. The original form of the electron microscope. It is produced two-dimensional, black and white images and magnifies objects up to 200,000 times. Second is the scanning electron microscope. Relies on interaction at the surface rather than the transmission. It can magnify bulk samples with a greater depth of view, so the image produced represents the 3D structure of the sample, but the image is still only black and white. Generally, it can magnify the object 10,000 times. Scanning acoustic microscopy or SAM, it consists of interpreting the action of a sound wave sent through a specimen. Sound wave of a specific frequency travels through the specimen a portion of its reflected back every time it is an interface within the material. It's also used to study living cells attached to another surface, such as cancer cells, artery plaque, and bacterial biofilms that fall equipment. Next, the scanning probe microscope. The scanning probe microscope was developed in the 1980s by the Swiss scientists Dr. Gerd Binig and Dr. Henrik Rohrer. It is used to study the molecular and atomic shape of organisms on a nanoscale. A physical probe is used to scan back and forth over the surface of a sample. A computer that gathers data that are used to generate an image of the surface. It can also be used to determine the variation in temperature inside the cell, as well as its chemical properties. Scanning tunneling microscopy. Tungsten probe scan produce an image revealing the bumps and depression of the atoms on the surface of the specimen. Resolving power is much greater than an electron microscope. It is used to provide incredibly detailed views of molecules such as DNA.